Thank you very much. It's truly an honor and a privilege to be here today, and so exciting to have the second Saskatchewan conference on PANS Pandas. I want to ask you to give another round of applause to Melissa, Robert, to all of the other parents that made this happen today. Melissa actually already did all my job for me, so thank you. I need to start with my disclosures. For the first time, I guess now that I've retired from the NIH, I do have a potential disclosure, and that is the fact that after several years of two very testy tech transfer offices debates, NIH was added to the Cunningham panel as a co-inventor, and so there may be royalties coming from that. I don't think it's going to influence what I will say about that, but I'll try to remind you again that there might be a bias there. I have no other financial or professional conflicts, and particularly now that I'm a scientist emerita instead of a full-time employee, I absolutely do not speak for the NIH or the US federal government. They would want you to know that. I thought we'd start with some definitions, and I will make my slides available. We'll take out any of the videos or anything that identifies patients, but the rest is available, so just feel free to relax and you can get details later. Because what I really want to have you take home today is a sense of, oh, that child that I saw in my clinic 3 weeks ago, where the mom had just presented a history of a change in behavior. He's just not acting like himself. Those are the kids that we want recognized early and steps taken to make sure that they don't have an infection, that they don't have another trigger for their psychiatric and behavioral problems. And if that happens often enough, then in a few years we won't have to talk about PANDAS or PANS. We will still have to talk about obsessive compulsive disorder. It happens in about 1 in 50 adolescents and young adults, and about 1 in 100 children prepubertally. So it's not uncommon. Despite that, you probably don't know about it in those children because it's a disorder of rational irrationality. The kids know that their forced behaviors and their worrisome thoughts don't make any sense, and so they try to hide them as much as possible. And it's been called a disorder of secrecy, or it's also known as the doubting disease, where they just can't be sure that what they see, what they feel, what they perceive is real. And as a result, particularly in young children, they constantly ask for reassurance. Mommy, am I okay? Mommy, did I touch that? Mommy, did I maybe make that child sick? Mommy, how do you know the sky is blue? And as you heard from Melissa, sometimes you have to answer that question in exactly the same way, and suddenly you have a family that's completely enmeshed with this disorder. Tics and Tourette syndrome are actually just the same thing. This is one reason the neurologists don't like me very much, but having studied obsessive compulsive disorder and Tourette syndrome now for a very long time, it's become increasingly clear that a simple compulsion and a complex tic are completely indistinguishable. A child with Tourette syndrome will feel a premonitory urge, feel a drive to perform that particular behavior, to do that vocalization. They can suppress that, just as a child with a compulsion can suppress the need to do that compulsive behavior. And as you heard, they can make it through the school day because it would kill them for their friends to know that they had this crazy disorder, and then they get home and everything falls apart. PANS, on the other hand, is a syndrome, and I'm going to go into that in more detail in a moment. But the acronym is just Pediatric Acute Onset Neuropsychiatric Syndrome. You might also see it as Childhood Acute Onset Neuropsychiatric Syndrome. Long story behind that, they're the same thing. Don't worry about whether it's CANS or PANS. It really doesn't make a difference. The acuity of onset is the secret here. And PANDAS was actually first described, but it is a subset of PANS. So about 1/2 of prepubertal children will have an abrupt onset, will have some of the other markers that we associate with pediatric acute onset neuropsychiatric syndrome. And of that 1/2 of the 1%, somewhere between 1/4 and 3/4, completely unknown fraction, will also meet criteria for PANDAS in that their symptoms were triggered by strep infection. Why am I making a distinction between syndrome of PANS and disorder of PANDAS? because it really does matter. And when you look at the controversy in the medical literature, it really comes down to the fact that the naysayers have always confused these two and associated requirements for a disorder to the behavioral description. In the world that I walk in, which is sort of developmental behavioral pediatrics, halfway between pediatrics and child psychiatry, we deal with both the DSM and the ICD codes. ICD codes disorders and diseases, DSM codes behavioral syndromes, or psychiatric disorders. PANS as a syndrome is a clinical description of a complex set of symptoms that always go together in the same way. And you can recognize them once you know what that pattern is. It's a little bit like looking up in the night sky and being able to recognize the Big Dipper because somebody taught you that those stars together are Ursa Major. PANDAS, on the other hand, is actually a disorder because we have a cause, we have a disease mechanism, we have specific treatments. Now, when you read the literature, the treatments that have been found to be effective for PANDAS have been applied to PANS, in large part because it's oftentimes difficult to identify that inciting strep infection. We can't make sure the child fits the criteria for PANDAS. And just now that we have an additional decade of experience, we're beginning to learn that you can treat most cases of PANS the same way as you could PANDAS. But please do, I know I'm going to mix them up all the time, and I'm going to talk about PANS/PANDAS as if they're the same thing, but this is your reminder that they're actually not. PANS is a behavioral syndrome. PANDAS is a disorder. Clinical criteria define both of them. So they're both made on a clinical basis. For PANS, there are just 3 criteria: the very abrupt onset of obsessive compulsive symptomatology or eating restrictions, as you heard, suddenly having picky eating. Some children refuse to take in any fluids. Oftentimes, this is because they have a fear that the food or the fluid is contaminated, but many times it's actually just a fear of gaining weight, a fear of being too fat, or frankly, they've just completely lost their ability to take in nutrition. At the same time, that child has to have other behavioral abnormalities start. And here's a list. We're going to go through this in great detail later, but suffice it to say that you have to have at least 2 of 7, and most children have 4 or 5 categories. So it's very clear that the syndrome, the behavioral presentation of PANS, is a child who falls apart in multiple domains. They not only have OCD and eating restrictions, but they're anxious, they're cognitively impaired, their handwriting is deteriorated, they're wetting the bed, they're having trouble sleeping, all the things you saw in those wonderful, wonderful examples that Melissa shared with us this morning. And third, and very important, another cause of controversy, but that's okay. Symptoms are not better explained by another medical disorder. Unfortunately, the insurance companies took advantage of this third criterion, and they deny treatment on the basis of the fact that you can't simultaneously have a diagnosis of PANS and autoimmune encephalitis. We need to make it clear that it is a currently defined form of autoimmune encephalitis, because as you're going to hear me argue at the end of the talk this morning, we believe that PANDAS is autoimmune encephalitis. So we've taken out that example and just left it as Sydenham-Chorea or Tourette's, or something where in Sydenham-Chorea there's a very defined and prescribed plan of treatment, and we don't want people to have missed that potential, forget to look at the heart in a child who presents with these things. In PANDAS, it was all focused on the strep. It was from the very beginning, and the criteria for PANDAS were actually written as criteria to define a research group of subjects studied at the National Institute of Mental Health in the 1980s and early 1990s, in which we were looking for a medical model of obsessive compulsive disorder. And because of that, we required at least 2 episodes of these behaviors that you heard this morning, both of them triggered by a known strep infection. We wanted to make sure that we got the purest group of patients. As these criteria have become used in the clinics, what clinicians are doing, and is completely appropriate now, is if you have a single strep-triggered episode, you can make a diagnosis of PANDAS. We also focused on the neurologic manifestations. I'll show you videos of the choreiform movements, but the fine piano playing movements, particularly during periods of symptom exacerbation, it also helped to define pandas and to separate it from other kinds of OCD. That distinction can be quite difficult to make. Again, you have to have the luxury of time to take a complete history, or as you heard some suggestions this morning, write out that summary, punctuate it with visual examples, and most importantly, remind the clinician of all the times they've seen that child when they've behaved perfectly appropriately for their age and developmental stage, and they haven't had these complex set of complaints. Because that's really the only distinction, is the acuity of onset and the fact that in PANS, symptoms of OCD or eating restrictions are accompanied by all these other symptomatology. The interventions for OCD in PANS pandas and OCD that's not PANS pandas are exactly the same, but the piece that you have to add in PANS is that you need to treat that entire complex of what's causing those symptoms. And so you'll hear more about the use of antibiotics and immune therapies. I'm going to give you a brain slide just to prove that I am a NIH neuroscientist. But this was a study done on some of our patients by Dr. B.J. Casey, a neuropsychologist who's now at Yale University. And what Dr. Casey was able to document was that in children with pandas, there's actually activation on fMRI in the orbital frontal striatal thalamal cortical circuit. This was so cool, at least to those of us who are complete NIH nerds, because that was the circuitry that had been predicted back in 1976 to be involved in obsessive compulsive disorder. And what is supposed to happen is that the orbital frontal cortex, the thinking part of the brain, initiates a thought. The basal ganglia is the gatekeeper and determines whether that thought is relevant, important, or whatever, and either lets it through and then positive feedback, or it stops it. In children with PANS pandas, that gatekeeper, the basal ganglia, appear to not be functioning properly, and so those thoughts get stuck, and you get in a do loop, just like the old computers. And activation can happen inappropriately and be maintained excessively long time. Well, as you saw, that was in 2002, so that was almost 20 years of work to document that. And more importantly, it was a very small study. So the relevance to all of PANS pandas, or even all of childhood onset OCD, is not well known. But what we do know is that this cohort of patients is going to give us a window into that brain dysfunction and obsessive compulsive disorder. As we're learning more about what those antibody-antigen interactions are, we're going to be able to design new and better medications to treat the disorders and particularly the symptoms, as well as actually getting at the heart cause of the symptomatology. But this is only a fraction of the children. Remember, I told you it was less than half of a half. So less than 1/4 of children with obsessive compulsive disorder are going to end up having also a PANS pandas diagnosis. Frankly, the most important diagnosis to make is that diagnosis of obsessive compulsive disorder, helping that child to recognize that their symptoms are senseless, that you've seen them before, that they are not something that is a unique symptom of craziness in that poor child, and that you can help them is tremendously useful. Within the PANS group, within children who have an unusually abrupt onset of symptoms, we sort of divide it just for the sake of convenience into two different pathways. One is infectious triggers, which were actually described about 5 years before the PANDAS cohort was described by my group, and A.J. Allen was the first author of that paper. And what we were able to document was that not just strep, but influenza, particularly the flu that we get every winter, varicella or chickenpox, and other infections. And more recently, our colleagues have shown that mycoplasma can also trigger symptomatology, and some have contended that Lyme disease and some of its associated infections can also trigger symptoms. In general, though, I believe that Lyme is not a common trigger of a PANS picture, mostly because of the difference in the way the disease works in Lyme disease, and that is that it's much slower, more indolent. You would actually probably have more difficulty separating a Lyme obsessive compulsive disorder from a non-Lyme OCD, not from PANS pandas, just because of the acuity of onset. Well, we focused on group A strep in large part because we were going to use rheumatic fever and its neurologic manifestation, which is Sydenham chorea, as our prototype disorder. Well, remember I told you that in the 1980s, we were looking for this medical model based on this orbital frontal basal ganglia thalamocortical circuitry. We looked at everything from Huntington's chorea to Sagawa's dystonia, and Dr. Rappaport and Dr. Mike Rutter in London were really the only two in the world who were thinking about obsessive compulsive disorder as a brain-based disorder. Everybody else was focusing on this as a childhood response to punitive toilet training and harsh parenting practices. These are the same people that blamed the mom, the refrigerator mom, for autism. So we've come a long way, not far enough, but at least we're not back in the dark ages. Because of the way that circuitry went, and because of Dr. Rappaport's knowledge of the literature, Sydenham chorea was chosen because it actually impacts the basal ganglia, and here are some old pathologic slides as well as some MRIs and CAT scans showing that the basal ganglia is either been destroyed by the inflammation or that it is swollen and inflamed at the time. Sydenham chorea is the sort of colloquial term for the neurologic manifestation of acute rheumatic fever. It used to be known as St. Vitus's dance. And that makes sense because the children have these involuntary movements, chorioathetoid, writhing, snake-like movements that come on top of everything. So if they reach for this tape, everything would be moving about and writhing about as they try to get the tape. Or if you ask them to touch their nose, that's a very difficult thing for them to do. It was difficult to prove that Sydenham chorea was part of rheumatic fever, and in fact, it wasn't added to the rheumatic fever family until shortly before I started at the NIH, in large part because the lag time between strep infection that incites that choreic behavior and the onset of the illness was so long. We still don't know what that intervening element is, but it's been shown in many, many studies that the child would develop the heart disease within 3 weeks of acquiring the strep infection, but the Sydenham chorea could happen several months later. The basal ganglia inflammation is known to be caused by cross-reactive antibodies, and Dr. Cunningham of the Cunningham panel has done a lot of that work, as well as pathological reports from the 1950s and work done by Husby and his colleagues in 1976. What they documented was that antibodies recognizing things on the strep cell wall also recognized things on the neuronal tissue. And that cross-reactivity is totally the fault of the strep bacteria, which puts antigens, which puts proteins and glycoproteins on its cell wall to mimic the human host. I was telling the reporter that this to me is sort of Harry Potter's invisibility cloak, where the strep just gets under their invisibility cloak of putting things on it that look just like the outside. And unfortunately, just like in the movies, when the edge of the cloak would come up where you knew where Harry and Hermione were, the immune system figures out that these are foreign proteins, sets up an immune response against them. And then unfortunately, those same antibodies can cross-react with the human tissue, if they can get access to it. And that was one of the biggest questions in pandas, was why would these antibodies that are in the serum and are have the potential to attack brain cells be allowed into the protective space of the brain? Work done by Dratan Agaliu, Pat Cleary, and others has given us a gorgeous model for how this happens. We know that the strep bacteria can produce exotoxins that actually open up the blood-brain barrier in the acute phase. But later, after recurrent strep infections, these same strep bacteria can trigger the production of T cells, helper immune cells, that can traffic into the central nervous system. And when that happens, you actually set up a neuroinflammatory pattern that is independent of any infectious trigger. So as we heard this morning, sometimes you find a strep, sometimes you don't. That certainly contributed to the controversy. It's also contributed to our clinical conundrum of, are different antibiotic going to help this child, or is it time to move to steroids or something else? And we're going to talk this evening about the importance of the consortium, or I guess tomorrow morning, and that's one reason that the consortium is so important. But I better get back on track, or we'll be here all through lunch. So back on track. Here's the model of etiopathogenesis for strep, and I actually just told you most of this without the benefit of the graphic, so I apologize. Certain strains of group A strep, about 10 to 15 of the 120 or more Lancefield strains of strep were described as rheumatogenic, had the property to produce rheumatic fever. And we think that it's a minority of strep that also cause pandas, fortunately. In the genetically susceptible host, you heard a beautiful example of how 3 children in the same family could be affected. Cheadle, when he was studying acute rheumatic fever in the tenements of New York and Chicago, described 10 of 11 children having rheumatic heart disease from the same scarlet fever epidemic, or the mom who had 8 of her 9 children in the lying-in hospital at the same time. So there's definitely a genetic susceptibility to rheumatic fever. We knew from studies that Dr. Rappaport and others had done in the 80s that there's a genetic susceptibility to obsessive compulsive disorder within families. If one child has OCD, their siblings and their parents have about a 1 in 5 incidence of having obsessive compulsive disorder, so about 20 times higher than expected in the population. And that genetic susceptibility in the child with pandas seems to come from both sides, so that you inherit the susceptibility to obsessive compulsive disorder or Tourette syndrome, and you also inherit that susceptibility to this exaggerated immune response to strep or other autoimmune phenomenon. And we call it an autoimmune response, but in my simplistic model, and certainly in rheumatic fever, we know that it's actually a misdirected immune response, and that the child's immune system may be completely healthy, that it's just temporary lost tolerance because of these tricky strep. And if we can get them back into that appropriate recognition of self versus foreign, then we have an opportunity to intervene. If we miss that and the central neuroinflammation begins, then we're left with the child with the chronic progressive or chronic static symptomatology that Dr. Tienemann will talk to you more about later. Well, just to nail this shut, so we can move on from Sydenham chorea, I just wanted to remind you that Sydenham, like pandas, is a diagnosis of exclusion. The differential diagnosis for Sydenham chorea is quite long, and those individuals have to not have other things that could be causing their chorea. You do a very complex and complete workup on the child, and you're left with no other explanation than maybe they had a strep, or if you do find rheumatic heart disease, then you're more sure that the child has Sydenham chorea. And what you're looking for is these writhing chorioathetoid movements. This first child I'm going to show you is a 5-year-old who looks like she might just be fidgety, but if you watch her closely enough, you'll see if she goes to shake her head, other things wiggle and twitch on her. In a moment, she's going to demonstrate for us a classic milkmaid's grip, which is caused by a failure to be able to maintain a tetanic contraction or maintain a grasp. So they literally milk your hand as they're holding onto it. Is she still going? I don't know why, it seems to be going unusually slowly. Here's the milkmaid's grip. So you see her grab it, let go, and the parents the first thing they might notice actually is they're holding the child's hand, and it just feels odd to hold this child's hand. Well, in a second, she's going to show you her mouth. She has a failure to maintain protrusion of her tongue. In extreme cases, it might be called snake-like tongue, but I'm going to start this other one at the same time. There's her snake-like tongue. And as she finishes up, we're going to watch this 12-year-old on the right who has a form of Sydenham chorea known as chorea mole, or the paralytic form of Sydenham, where the prominent symptom is actually muscular weakness and a failure to maintain tone. In extreme cases, the child completely bedbound, unable to lift their head up off of the pillow. You can see that she actually does have chorioathetoid movements. Her toes curl up with every step that she takes, and if we were watching her hands, they would be having overflow as well. Well, when you went back to these rich clinical descriptions from the early 1900s, what you saw was in Sydenham chorea, 95% of the patients had just debilitating emotional lability. Rapid, unprovoked mood swings, they were suicidal, there were many, many historical links, and because we were studying OCD, we focused on that, but when you look back at those old reports, the entire syndrome of PANS is there. The child had a refusal to eat because she thought the food was contaminated. This, case number 4 actually had to be tube fed because her eating restrictions were so severe. Other ones in this paper, they talked about psychotic symptoms. I think you see psychotic symptoms. In case 4, she saw mice and moving objects in her room, saw a man climbing through the window, many of the same kind of behaviors. In the case of Sydenham's, during wakefulness as well as during sleep. And I love these historical reports because you read on in this report, and case number 4, it's going through this very long description, and then it says, between these episodes, she was quite a delightful child. I mentioned that the relationship between Sydenham chorea and OCD was actually made in the 1800s by Sir William Osler, who's sort of the American father of modern medicine. He seems to have described everything. But in the 1950s and 60s, 3 separate investigators looked and found that obsessional neurosis, or what we would now call OCD, was much more frequently occurring among children with Sydenham chorea and adults who had had Sydenham's during childhood. We did one retrospective study at the NIH where we compared children who had Sydenham chorea against those who had other kinds of rheumatic fever, usually rheumatic heart disease without chorea, and found those that only had the anti-heart antibodies and heart disease did not have any OCD, whereas 2/3 of the children with Sydenham chorea had a new onset of obsessive compulsive symptomatology. We then did prospective studies at the NIH and just investigated these children from stem to stern and found that that rate stayed about the same. It was confirmed in a clinic in Brazil where they had an unselected group of patients, and there they unfortunately had a third world population who often was unable to get to appropriate prophylaxis, and so the children had multiple episodes of Sydenham chorea, and they found that the incidence of OCD actually increased to 100% after the 3rd episode. Very important for us doing the research and for this to be a medical model, in all cases, the obsessive compulsive symptoms were the first things to start. And they started 2 to 4 weeks before this child ever had these writhing movements. Remember, I told you that this was the era when we thought punitive toilet training was the cause of OCD. So if these children had suddenly been neurologically debilitated and began having obsessive compulsive symptoms, the natural construct would have been to think they were just trying to regain control of some aspect of their life. But in actual fact, they started worrying that their sister would get sick, the little sister they didn't particularly like, or that they had germs on their hands, or that their hands smelled funny. And the first case of Pandas was actually sent to us for a study that we were doing on Sydenham chorea. He was the 3rd of 3 boys, and his oldest brother had Tourette's syndrome. His mom was a medical technologist, and this was in the days when if you were a med tech or a pediatrician and you thought your child was sick, you could just bring home a swab their throat, take it back to the lab, culture it, and then when you went to the pediatrician the next day, you already knew the child had a positive throat culture. Now we have minute clinics and rapid strep tests, so we don't have to do that anymore. But this mom had already made the observation that every time her oldest son's tics became much more prominent, he would, within a day or 2, end up having to miss school for a strep pharyngitis. So she had already learned that if she saw his tics increase, to swab his throat, would treat it with antibiotics, and the tics would go back down. So she's the one that actually discovered the relationship. We came to it through extrapolation of Sydenham chorea, and this patient was the perfect link because he had these wild choreoathetoid movements, and he was sent to us as a Sydenham chorea because of the presence of these bizarre movements and dysarthria. Well, at the NIH, I told you we worked them up from stem to stern. Our initial intake interview is 4 hours. We do split it, give them a little bit of break, but it's a long period of time. And what we noticed was that this child was dysarthric because he wasn't swallowing his spit. And when confronted, he said it was because he had to hold on to anything that had ever been part of him. He had terrible hoarding obsessions and compulsions, and he would spit into a cup and then line those cups up at home. Sorry, I don't like spit. He also would clear out our waiting room of every magazine, newspaper, anything that he saw, put it in a brown bag he carried with him and take it home. These movements, as you saw in the 2 girls, you couldn't predict where it was going to be the next time, depending on what they were doing. In this boy, it was always exactly the same. and this arm went out, and this one came up. And what he was doing, he had an obsessional thought, and he was flinging them away by doing this particular motion, and it was clearly a compulsion. He had a positive throat culture. When we swabbed his throat, we treated him with antibiotics, and his symptoms slowly improved. The key came that when he got strep a few months later, everything came back. We treated him with antibiotics again, and they again resolved. So he was one of the first. We ultimately accumulated the series of 50 patients that were described in that article in 1998, and we sort of entered two decades of discovery from the mid-'80s to the mid-2000s, where the NIMH group and a few others across the country, and eventually Italy and other places across the world, were just quietly making these connections between the Sydenham chorea and OCD, and using it as a medical model for understanding obsessive-compulsive disorder. Those 2 decades led to 5 lines of research showing that strep was the etiologic agent in pandas. So this is a disorder with a known disease mechanism and a likely pathogenic trigger. Group A strep was chosen again for convenience sake because we had that medical model in rheumatic fever, but we actually, in our studies of pandas, have been able to go beyond rheumatic heart disease because they still haven't been able to document the specific antibody-antigen reaction that gives you rheumatic carditis or what other immune factors are involved. The first thing was clinical observations. A number of epidemiologic studies, particularly recently, have been done showing that if a child has strep, they have a much higher chance of having OCD or tic disorder. And in fact, Dr. Tanya Murphy, who was here at our last meeting in Saskatoon, Dr. Murphy and her group were able to go into an elementary school in Putnam County, Florida, and document that when children had recurrent strep, their chances of having choreiform movements, other adventitious movements, and problematic behaviors was markedly increased. And she did a one-to-one correlation because those children were being examined and surveyed for rheumatic fever prophylaxis with monthly throat cultures. So positive throat culture was actually known the day after those observations had been made that the child had OCD-like behaviors or choreiform movements. Treatment of the strep was able to reduce these OCD and tic exacerbations. Paper in 2002 from Picacchiero, and at his pediatric practice in New York State, they documented in 12 children that 8 of them had a strep trigger for their obsessive-compulsive symptomatology. And then probably the most definitive thing in rheumatic heart disease was the fact that if you were able to prevent future strep infections, you could prevent future episodes of rheumatic heart disease. And so we took that same model in pandas. Now, I'm so glad that Melissa's already spoken with us this morning because you've already heard how hard it is to keep strep out of a family. And in our first study of antibiotic prophylaxis for pandas, we did a really good job of proving that. 14 of the 35 infections that happened in that group of children actually happened to the children who were taking penicillin in the blinded study. So we went back and we assumed that penicillin wasn't going to do a very good job of preventing strep, and that therefore, if we could completely eradicate strep, we might see a difference. We actually compared azithromycin, which only has to be given once a week for prophylaxis, versus penicillin, which has to be given very religiously every 12 hours in order to keep the half-life high enough that it actually can do its job of preventing strep infections. And this was a 1-year-long trial with monthly visits for the sort of severity or presence of their obsessive-compulsive symptoms, other neuropsychiatric symptomatology, as well as a throat culture and a check of their titers. What we discovered was if you give penicillin every 12 hours, which we were doing, they had blister packs, we had phone calls as a reminder, they kept a medication diary, the adherence in that study was complete, and we were able to completely eradicate strep in both cohorts. And we also markedly reduced their number of exacerbations from 2 per subject down to 0.8. And more importantly, the number of symptom-free months was quite increased for the children during the period in which they were on antibiotic prophylaxis. So that was actually done, that study was done as a demonstration that strep could be an etiologic factor. It led to, obviously, the clinical application of antibiotic prophylaxis being one tool that we can use to help intervene in this disease. But the most definitive evidence that strep is the etiologic factor, the causative factor for pandas, came from animal model work. With the study by Dilipan, and here the senior authors were Ratan Agalyu, who's now at Columbia, and Pat Cleary, who's at the University of Minnesota, and this is the one I hinted at before, where what they did was they took a group of mice and they gave them 4 strep infections, a strep infection in their nose once a week for 4 weeks, and at the end of that time period, the animals were sacrificed, and what they documented was that these mice had T cells that also recognized the strep cells, and those T cells were trafficking along the olfactory nerve into the base of the brain. Well, it was very important that they use the intranasal model. They actually tried to do it sub-Q. They tried to do it intravenous. It's only that intranasal model, and they actually infected what's called the NALT, the nasal-associated lymphoid tissue. In a human, the equivalent would be our adenoids. And so one of the things you heard was the tonsillectomies can sometimes help these children. We've begun asking, is it actually the tonsillectomy, which certainly could be a source of an occult strep infection, or maybe is it the fact that usually when you do tonsillectomy, you also do adenoidectomy and remove that source next to the cribriform plate. Human data on that are not going to come. It's really not a good experiment to do in children. So we'll continue to work with the animals and try and find the answer to that. In the meantime, the official recommendation of the U.S. Consortium is not tonsillectomy unless the child has other reasons for having a tonsillectomy done. Well, my colleague, Madeline Cunningham, for many years, even longer than I have, has been studying acute rheumatic heart disease, and we convinced her to look at Sydenham chorea as well as pandas, and what she found was the presence of these cross-reactive antibodies. We reported this in the 1990s, and it was initially met with actually not much recognition at all until we began challenging the fact that when Singer and Curlin and others attempted to replicate our pandas cohort, they chose a group of children who didn't fully meet the pandas criteria, and they were actually probably studying children with non-pandas Tourette's OCD. The reason this is so offensive is because we already knew from this paper published in 2003 that those children would not have the antibodies present. In fact, they were our contrast group. So in Sydenham chorea and pandas, you see similar staining, and what these slides are showing is that antibodies that recognize the strep are now also adhering to neuronal tissue, here taken from the caudate and putamen, so the basal ganglia tissue that we knew to be involved in OCD and Sydenham chorea. The healthy children did not have those antibodies present in their blood, and neither did children with non-pandas OCD or Tourette syndrome. So in my opinion, the controversy was contrived, it was false, and this slide should be sufficient to end the controversy. I know that that's a foolhardy wish, so I'm going to hope to convince you by the end of this time together that the controversy does need to die, and fortunately, I just found out that the American Academy of Pediatrics has chosen PANS Pandas as one of its 2 things to do a special panel for this next year. So hopefully by this time next year, the AAP, the American Academy of Pediatrics, will have a statement. I've been told that they could find the pandas doesn't exist. I think the chances of that are small. Those antibodies cause these children's symptoms because they're actually reacting with dopaminergic receptors, D2 and D1 receptors within the brain. Also, anti-lysoganglioside, and the lysogangliosides are not only a very frequent component of the strep cell wall, but they're actually a part of the architecture within that holds up the brain. And more recently, Chris Pittenger and his colleagues at Yale have found that the antibodies also recognize cholinergic interneurons, some of the intervening structure, again explaining how one child's symptoms could be so different than another child's. It's because the antibodies, the inflammation is caused against the structure rather than even necessarily the neurons. And these antibodies are biologically active. What this graph shows is that in children with Sydenham chorea, their serum increases calcium calmodulin kinase 2 activity by over 200%. Pandas is intermediate, about 150%, and the healthy controls, you'll notice that the highest level there is 130. So Dr. Cunningham set the threshold at 140 for the Cunningham panel test. If the CAM kinase is elevated, it means that serum has the capacity to induce cell signaling within neuronal cells. The animal models, in addition to the elegant experiment looking at strep by Dilipan, we also had a number of different animal models, in fact 6 models from 5 different groups around the world, all of which have documented that the infusion of serum or even CSF from pandas patients or from Sydenham chorea patients has the capacity to produce behavioral abnormalities in a group of mice. Then when the serum from those affected mice is taken and transferred to a group of naive mice, those mice also document the abnormalities. That is Witebsky's 4th proof that a disorder is immune-mediated, the fact that the disorder is transferable from one affected animal to another. Well, those 2 decades of discovery were followed by at least a decade of controversy, and I've told you, obviously, my side of this argument. Frankly, it's getting really old. An unnamed scientist with their magnifying glass looking in the bush when the pandas is hanging up like the piñata was that the kids were fighting against. To be fair, we have thought about it, and we've actually learned a lot from the controversy. I think if the controversy hadn't been so ugly, we probably wouldn't have worked as hard as we have to prove the disease mechanism or to try and disprove the disease mechanism, and we've really learned a lot over this time. And as a general pediatrician, before I came to the NIH, I know that strep infections are common, and so if somebody came to me at that point in time and said, oh, that child's washing his hands and having new separation fears and sleep difficulties, you should check a throat culture, I would have looked at them like they had 2 heads. Because 80 to 90% of grade school age children are going to get a strep infection every year, some of them many times. So there was a great chance that this was a true unrelated finding, that it's true the child had a strep and it's true that they had an abrupt onset of symptomatology, but those 2 things had absolutely nothing to do with each other. The other monkey wrench here is the fact that 5% of children described as strep carriers. They aren't bothered by the strep. They're the ones that are giving it to everybody else in the house or in the classroom. The strep just lives perfectly happy in their little oropharynx and is available to be spread to other susceptible individuals. So it could have been a true, unrelated finding, or it could have just been a spurious finding. And in fact, when we studied our group of 50 children, what we found was that if you looked at their titers, and we would now use a battery much more complete because we know that the anti-DNase-B and the ASO, even if done in a child with a proven positive throat culture, only increases in about 50 to 55% of the kids. So you have almost a 50-50 chance of missing the strep infection by doing titers. But in these 2 cases, what you are supposed to be able to see is in that child with pandas, when the strep titers, anti-strep titers go up, their symptoms are already high. And as those titers wane, the symptoms come down. It's not one-to-one like that, and I do not want you to leave here thinking strep antibodies equals pandas, because it's very clear that these antibodies may be necessary, but they are not sufficient to cause this syndrome. And so therefore, you have to back away from it a little bit, and we don't do titers routinely on our children with pandas. In the non-pandas child, you have just as good a chance of getting a positive titer if you measured it there, and at that point, their symptoms, documented in the red line, were also quite high. But you see when the titer comes back down, the child's symptoms actually are increasing, and there's no relationship at all. The other thing I've already told you is the fact that we already knew from the early 1990s that strep is not the only infection to trigger these symptoms. So not finding a strep may not be meaningful. The other reason that PANDAS was considered so controversial was some of the leading movement disorder experts in the United States, Jonathan Mink, Harvey Singer, Roger Curlin, and Don Gilbert, just weren't seeing it in their Tourette syndrome clinic. There's one paper by Dr. Singer, which he likes to forget that he ever authored, which was when Henrietta Leonard and I went to his clinic, we spent time in the waiting room interviewing those families that were willing to speak with us, and then gave those results to him, and Harvey wrote how 10% of the children in his clinic met criteria for PANDAS. He did not know that at all, because what he was focusing on was what was being presented to him in front of him, and that was the fact the child was very troubled by their tic disorder, and he was treating the tics. Despite that, there have been significant arguments, and in fact, we've been told that we missed Sydenham chorea, that I'm just a pediatrician, so I should have had a neurologist. Again, they ignore the fact that all of our children were evaluated by our colleagues in the National Institute of Neurologic Disorders and Stroke, Dr. Mark Hallett, who's in charge of their movement disorder clinic, and he does know chorea, even if I don't. But I think that I can make you a diagnostician just with the next 2 slides. And the reason this is important is because one of the arguments that Gilbert uses even now in his 2019 paper is that the only difference between chorea and these so-called choreiform movements is the amplitude. Now, my brother's kind of a smart aleck, and so when I told him this, he said, well, yeah, Sue, the only difference between a speed bump and Mount Everest is amplitude. So he's right. I think there's more of a difference than that. And here's a child who has, I'll show him, you can actually make the diagnosis for me. I'm not going to say, I want you to shout it out. What does he have? He has to put his arms up. What's his hand doing? Right? Can't hold still. So what does he have? Thank you, Sydenham chorea. He definitely has Sydenham chorea. The other way you could know, if I showed you all 29 examples, every time the examiner's standing within arm's reach of the child, they have Sydenham, because there's a good chance that one of those movements will make him fall over. Oh, I'm sorry, I need to go back, because the other thing you should have seen in this child was the fact that he has eye deviation. And so sometimes those eye movement tics are actually a subtle presentation of a choreotic movement. This child does not have Sydenham chorea, and they're not normal. Oops, I don't want volume. Thank you. Right? He's very still. His face is still, his arms are still, and the only thing that's moving is his fingers. Let me just play that part over again, because I want you to just notice how it just comes out of nothing. We asked him afterwards if he knew his fingers were moving. He really wasn't aware of it at all. Might also get some pronator drift if Tanya always having them put their hands up here. But probably the very best way to determine if they have choreiform movements present is actually due to stressed gaits. So you'd have them walk on their heels, walk on their toes, walk on the edge of their feet on a straight line on the floor. Heel-toe walking can sometimes bring it out, and what you see is that their hands begin writhing around and they have a lot of overflow to the hands. I do have, at the end, normal, but I promise you that any child over 6 years of age, if they are neurotypical and healthy, should be able to just hold their hands out. And if I had shown you this young boy 2 months later, he could indeed hold his hands straight out. Below the age of 6, choreiform movements are normal, and so that's actually kind of an exciting finding, because what it suggests is that these antibodies are releasing inhibition, which would make sense with these thoughts continuing inappropriately. All right, I'm going to speed up, because otherwise, I'm already over time. Is that right? How much? A few minutes. A few minutes. Okay. Well, you know how to diagnose this now, right? And we heard video this morning. The only thing I need to get you through is to PANS. And then Dr. Tienemann will be able to take over this afternoon with the diagnostic workup. Comorbid symptomatology, we don't have to linger on this because you've seen it all, except to say that it is constant. If a child comes in with an abrupt onset of OCD only, I would personally be worrying about some form of abuse, some other psychosocial trauma. and that the OCD is a manifestation of that. Even though I was poo-pooing the punitive toilet training construct, we have indeed, in our large OCD studies, seen girls in particular who presented with an abrupt onset of OCD, and both of those girls had been abused. And we're doing washing literally as an attempt to regain control. Behavioral regression is very prominent, separation anxiety in nearly all of the children, emotional ability, just as in Sydenham, Korea, 95%, somewhere between 95 and 100% of our children. They also have cognitive changes, hyperactivity, inattentiveness, hallucinations. This is a very, very important symptom, and maybe Dr. Tienemann can talk about it more this afternoon, because her group at Stanford is the one who has reported on the fact that when the child presents with either obsessive compulsive symptoms that have lost insight and have really gone to the point of being delusional concerns about germs and contamination, or frank hallucinations, like in those two children with Sydenham, Korea, where they were seeing something in their room that wasn't there. Our children have Capgras syndrome, where they don't recognize their parents and loved ones as familiar figures. They've had the Alice in Wonderland, where they're looking at something and suddenly watch it shrink. You can imagine how frightening these symptoms are to the child, and particularly for young children who don't have the capacity to tell us what's going on in their thoughts. The terror-stricken look, that deer in the headlights, these children are in a very hyper-aroused, hyper-vigilant state. They're literally in constant fight or flight mode, and they have two responses. They're crying this very primal scream. Sorry, Melissa. What we heard this morning is the panda's cry. It's the child who is so much angst, so much anxiety, that they are just beside themselves. Or they're going to react by fighting against you, rage attacks, destroying property, trying to destroy themselves. They're so, so angry with the disease. The pathognomonic sign for Sydenham chorea is actually that milkmaid's grip. When you find that, you say, oh, this kid has Sydenham's or has some form of chorea. In PANS pandas, our pathognomonic sign, sort of the telltale sign, is the presence of urinary changes. Urinary frequency, 20 times an hour, voiding, and immediately by the time they get to the bathroom door feeling like they have to void again, is probably because the internal and external sphincter are under different neurologic control. And what we think what happens is that they lose that sort of autonomic internal sphincter control of micturition, of urination, and so they do have urine in their urethra, and for particularly little boys will experience a feeling of needing to void again just because they have a drop or two of urine. They also start having daytime accidents, new onset of nighttime bedwetting, so secondary enuresis. Secondary enuresis and a behavioral symptom, swab that throat and go to the PANDAS Physicians Network and get the Dr. Tienemann's article on what to do with your first patient with PANS. Handwriting deteriorations is a great way to be able to make the diagnosis retrospectively. You saw that if you looked back at that child's school journals or their school papers, and then compared that against the medical records for not just that child, but their siblings, when there's strep in the home or in that child, handwriting deteriorates. Great sign that that child is in the PANDAS cohort. Sensory hypersensitivity or insensitivity, these kids look like they are encephalopathic. They have light sensitivity, sensitivity to sounds. They'll come in wearing earphones, almost like the children with autism. Well, that constellation of symptoms can go anywhere, and it can be very difficult to predict, although with the experience that we're now getting by sharing cases across the United States, the consortium is beginning to feel like there are probably 3 or 4 major ways that this disorder presents, and that some children tend to be more bothered by the irritability, the rage, the impulsivity, the behavioral regression. Other children are more tortured by the internal psychiatric symptoms of anxiety, depression, and obsessional fears. When you see that abrupt or lightning-like onset of symptoms in the combination, you're thinking about PANS pandas. You've already seen an example of the motoric dysfunction. Here's another one in which the child is very able to write beautifully, stay on the lines, and loses that ability to form letters neatly. Criteria require you to have 2 of 7 symptoms. In actual fact, most children have 5. And they have cognitive, emotional, behavioral, somatic, and sensory motor abnormalities that tells you that you have an encephalopathic process. We're going to skip through these quickly. Behavioral regression, begin acting like children. Disorders of executive functions, these do not appear to abate, at least in patients that we tested with Sydenham chorea. They retained some dysfunction of executive skills, as well as some visual spatial skills deficits. They have difficulty finding maps. Fortunately, we live today, and if you've got Google Maps, you don't have to worry. Ray asterisk, this is a sign actually of working memory. The children actually don't do as bad on recall of this complex figure as they do trying to copy it, and that's a sign of the visual spatial skills deficits where they're unable to make that one-to-one representation between the figure that they see on the left and their blank paper on the right that they're supposed to recreate the figure as they look at it. Unusual margin drift, I didn't get it in here, I apologize, but I finally have a clock showing the left-sided neglect. So we do believe that this margin drift, which again might be present in the school journals at that same time that the child has messy handwriting, indicates that the child has a left-sided neglect. That's actually a sign more of a true encephalopathic process that we might call delirium. Oops, I went backwards, I apologize. So the medical workup, I just want to get to the fact that we have begun to conceptualize of pandas as autoimmune encephalitis of the basal ganglia. And Dr. Russell Dale, who's now in Australia, deserves credit for making this connection, rebranding pandas, and not only will pandas disappear within the next decade if my hopes are met because we get rid of it, but it will disappear even more quickly because we'll start calling it AE basal ganglia. The reason is because autoimmune encephalitis is a complex set of psychiatric and neurologic symptoms. Antibody-positive cases, anti-NMDA, anti-GAD, anti-potassium channel, all the different antigens within the brain have been examined to see if they have antibodies against them. And the clinical presentation of those antibody-positive cases is really fascinating because it fits exactly what the presumed function of that neurotransmitter is. So when you're binding up the neurotransmitter and it can't do its job, surprise, surprise, you get symptoms of not having a functional antigen. Well, in children in particular, antibody-positive cases are just as common as antibody-negative cases are just as common as antibody-positive. You can't find an antibody against the brain, and you're left with sort of this confusing clinical picture, maybe some mild abnormalities on their spinal fluid, other things, and you end up treating the child as autoimmune encephalitis because if you wait for everything to manifest, you end up at a point that the child is going to be quite ill and it's much more difficult to treat them. So the Consortium for the Autoimmune Encephalitis Society has determined that the AE can be diagnosed on the basis of 2 of 3 things: clinical presentation, abnormalities of paraclinical studies or lab assays, or response to immune therapies. So basically, if a child comes in and you suspect they might have autoimmune encephalitis, you treat them as an autoimmune encephalitis and they get better, that's your proof that they were AE. Granted, it's a little bit circular argument because it may have been something else causing it, and that's why before you make a diagnosis of autoimmune encephalitis, you have to do a complex workup. You'll hear more about this afternoon. In pandas, we actually have this very complex clinical presentation with multiple domains being affected, but we also have paraclinical support. In a fraction of the children, you're going to see CSF pleocytosis, not necessarily an increase in the white cell count, but the presence of atypical lymphs and other markers of microglial activation, an abnormal IgG index, other things. So a spinal tap should be done, in my opinion, on every child whose panda symptoms are severe enough that they're in an emergency room. CSF and serum cross-reactive antibodies, we know from the work that we've done with Madeline Cunningham that both serum and CSF are positive during acute illness. When we treated the children with IVIG, retapped them, and if they were better, 6 weeks later, the titers were lower in both children with pandas and with Sydenham chorea. And MRI and PET have demonstrated basal ganglia inflammation, and we'll get to the EEG and polysomnography in a moment. MRI evidence of regional inflammation is only on a group basis. The chances of you finding the next slide in your patient population are frankly slim to none. So you just have to depend on the research results, which showed that there was increase in size in the caudate, putamen, and globus pallidus during acute illness in a group of 20-some children with pandas compared to controls. In the next case, who was one of those, we actually were able to see a 20% enlargement over expected size for his age, sex, and weight in a 14-year-old boy who had a very severe case of pandas contamination fears and exercising rituals. His first presentation. On his next presentation, when he had a second exacerbation, his primary symptom was actually eating restrictions and severe anorexia. He was treated with plasmapheresis. His symptoms completely abated, and he also had a return to normal of his caudate size. Harry Shigani and his colleagues, when he was at Wayne State University, used PET and a ligand that recognizes activated microglia, a sign of inflammation, and what they found was that there was inflammation within the caudate and lentiform nucleus in children with pandas, and in this case, this child was treated successfully with IVIG, rescanned, and those activated microglia were no longer present. Interestingly, in that study, presented as another cause for controversy by the neurologist, but to us it suggested that perhaps more of Tourette's is immune-based than we might have thought. Dr. Shigani actually found that Tourette's who did not have a PANS history also had activated microglia present in their lentiform nucleus. Not as widespread as seen in the pandas, and you could distinguish between the two, but definitely some inflammation there. Electroencephalography, when it's positive, is very helpful. It's only positive, though, in about 16% of the children. And in both Sydenham chorea and pandas, what you would see is nonspecific diffuse or focal slowing in about 7%, or epileptiform abnormalities. And there's some debate in the field whether epileptiform abnormalities are meaningful. We think that they are in these children because when they were studied again during convalescence, the findings were absent. So about 17% had abnormalities on their EEG. And in our study, where we didn't wire the children up as badly as Melissa's daughter, we found REM behavior disorder, REM sleep abnormalities in 86%. So if you have only one paraclinical study to do in a child with pandas, I think the sleep study is the way to go. And Dr. Tienemann may share the Stanford results with us later, but they found that they actually persist, particularly in children who have persistent symptomatology. In our cohort, those children who responded to treatment appeared to have disappearance of their symptoms following successful treatment. And then the final thing for AE was that you improve with treatment. And we are going to talk more about treatment tomorrow, so I'll just stop here and move right through this and just say, in my opinion, we have met all of the requirements for pandas to be considered a form of autoimmune encephalitis. You should be able to treat it on that basis using standard treatments for AE, which include immunomodulatory therapies, psychiatric and neuroleptic medications as needed to control symptoms, and in the case of an infectious triggered one, removal of the source of infection. And with that, I'll close and thank all my colleagues. Two of my favorites are here with us, and I'm so grateful to them for joining me in this conference and to all of you for inviting us. Thank you. Now that I'm the Chief Science Officer of the Pandas Physician Network, I'll just put in another pitch for the Pandas PPN. It's actually a wonderful, wonderful group. You also heard that the Pandas Network is incredibly helpful, but I personally, if I lived in Canada, would turn to the Facebook and some of the resources you have here in Canada, because you guys are at the forefront. International OCD Foundation has also now separated out a very Pandas-specific focus, and so they're improving their website in terms of information and availability. And a new group is forming in the United States that I'm hoping Saskatoon Pandas might be able to join once they're officially launched in July. So watch for those announcements, and we'll know more about it then. Thank you again.